Assalamu alaikum. Our topic for today is the antiviral agents, and in this topic we will discuss in details the viral cell and the structure of the viral cell and how it multiply and grow, and what are the possible and available antiviral agents and their mechanism of action on different types of viruses. So first of all, we all know that the virus or the viral cell, it is a non-cellular. So it cannot live, it cannot survive outside the host cell. It depends on the host cell in growth and multiplication. So this is very important issue. Another thing that the viral cell can invade almost all living organisms. So we have some viral types that invade bacterial cells. Other can infect plant or animal. And we have more than 400 types of viruses can infect human. How the viral cell can spread between individuals. So the major route of transmission is through air or the airborne by sneezing, coughing, such as flu virus, chickenpox, measles, mumps, viral pneumonia, rubella, and smallpox. Other types of viral cell can transmitted through insects by insect bites, such as the yellow fever. And the third possible method for transmission is through food and water. And we have the hepatitis A and E, poliomyelitis, and the viral gastroenteritis. And through decades, we have so many epidemic viral infections that cause casualties and millions of deaths all over the world. For example, smallpox that hit the Roman Empire during the 165 AD, and millions of people have died at that time. We have the flu virus that have been an pandemic in 1918 with 20 million deaths. We have the HIV, which is still a life-threatening viral infections, and with almost 30 millions died since 1980s. We have the dengue virus, we have SARS, Ebola, and nowadays we are challenging the coronavirus, which is the COVID-19, with more than 3 million cases all over the world. So it's very important to find a way, first of all, to prevent the infection, the viral infection. And we are talking here about vaccination. And the other thing, we should find a possible antiviral agents. And the most important thing in the antiviral agent, it should be highly selective on the viral cell. Another thing, it must have a broad spectrum of activity. Okay, so in general, the viral or the virus is a protein package transmitting a foreign nucleic acid between host cells. So it's not a real cell. It cannot grow outside the living cell. So it depends on the host cell for maturation, for multiplication, and for survival. And we are going to talk about this in a minute. According to the viral nucleic acid type, we can classify viral cell into major two types. That one containing the RNA as a nucleic acid it is called the RNA virus, which is more virulent and more resistant compared to the viral cell that containing the DNA as the main nucleic acid. So we have RNA virus and we have DNA virus. According to the chemical structure of the viral cell, as we said before, virus is a nucleic acid material packaged within a protein called capsid. So the nucleic acid, it's coated by a protein called capsid. As we can see here in the structure, we have the nucleic acid inside the capsid. And in most of the viral cell, we have an additional membrane which consists of different types of proteins and lipids. And the most important proteins are the glutamine and the neuroaminidase, in addition to some types of viral proteins inside the capsid. So this is in general the common structural features found in the viral cell. So the major important part is the capsid, which contain the nucleic acid inside. The mature viral cell is called virion. Okay. Regarding the chemical structure or the shape of the viral cell, we have some viral cell with a cubic shape, some of them with a helical shape, and the rest could have different shapes such as spherical, brick shape, ovoid, or pleomorphic. And according to the nomenclature of the viral cell, some of them will end with viridae, such as boxyridae, for example, and most of them will end with virus. So, for example, pox virus or polio virus, HIV virus, and so on. So either this way or this way. So this is the common nomenclature or naming the viral cell. As we can see here, this is the general structure of the viral envelope. As we can see in the core, we have the capsid with the nucleic acid and some important enzymes for multiplication inside the capsid. And we have the envelope, which containing the important glycoproteins, and these proteins are very important in the invasion process as well as in the immunogenicity of the viral cell. And this glycoproteins is the fingerprint for each viral cell. So most of the viral cells, they differ in the types of glycoproteins, types and shapes as well. Because of that, some vaccines will not work on one type of viral, but it will work on the other. It depends on the shape and the type of glycoprotein. Okay, let's just talk in brief about the life cycle of the virus, and we will start with the DNA virus. And here the nucleic acid inside is the DNA, and as we can see in the left side of the slide here, the virion will come and it will be adsorbed on the surface of the host cell by the means of glycoproteins. And as we can see here, it is very important for the attachment then we will end up with what is called the pinocytosis, or the penetration by fusion, that will expel the capsid into the host cell. After that, the DNA will come out from the capsid in order to be incorporated inside the host DNA. After that, the host cell will be divided and the DNA will be replicated along with the viral DNA. So we will end up with multiple copies from the viral DNA that will be encapsulated again by the nucleocapsid, and after that it will be budded, or it will be expelled outside the host cell, taking part of the host cell membrane with the different glycoprotein, and we will end up with a mature virion. This process definitely will kill the host cell, but it will help the virus to make a multiple copies from it. So let's just now talk about vaccination before start talking about the possible and the available antiviral agents in the market. Vaccine means to protect ourselves against the viral infections. And as we know, we have so many vaccines for viral infections, and we normally take these vaccines in the childhood, such as polio, measles, طبعا measles الحصبة, الممps اللي هي النكاف, smallpox الجدري, and the yellow fever. Okay, the first one who created vaccines is Edward Jenner in the 1800s year, and we started using vaccination all over the world, and especially for the DNA virus rather than RNA virus. So vaccine is still ineffective against so many viral infections, especially those with a rapidly mutating surface, especially the RNA virus. هذا النوع من الفيرال سيل بصير فيه ميوتيشن بشكل سريع، وخصوصا للسيرفس جلايكوبروتينز. فإذا أنت عملت vaccine for example flu virus this year, it will not work for the next year because the flu virus is mutating. So we will end up with new glycoproteins. We will end up with new surface proteins, so different antigenic characteristics. Another viral cell that still we don't have a vaccine for it is the HIV, the dengue virus, genital herpes, Ebola, and so many others, especially the RNA virus. Okay, and as we know about vaccination and how to prepare the vaccine, we will either give the patient the attenuated viral cell or a dead body. So let's just write this down. So in order to prepare the vaccine, you should have either the attenuated virus or weak virus, or we can administer the dead virus. So it's better to give the vaccine as attenuated virus, because in this type we still have the whole viral cell with glycoproteins which is the surface proteins intact, and the cell membrane is there, so the whole virion is there. So it's very important to have the whole viral cell in order for our immune system to produce antibodies, suitable antibodies for this viral cell. On the other hand, the dead virus, we might have different shape, different or damaged cell membrane. So this might not help in producing effective vaccines for the living viral cell. On the other hand, the attenuated virus, we cannot administer this viral cell to patients with low immunity, because in this case, this viral cell might infect this patient. So it's very important to have a good immunity before giving or administering the vaccine to these patients. فعشان هيك أي شخص بيجينا بده ياخذ الفاكسين، for example flu vaccine، وشعرنا إنه he has, for example, he has got infection, he has a flu, he is taking, for example, immunosuppressant drugs, so we cannot administer or give him or prescribe him a vaccine. Okay? So we'll just start talking about the antiviral agents, and we will continue this topic in the next lecture. Antiviral drugs, they are very useful if we have viral infections. So it is they are differ from the vaccination. Vaccines, it is not effective on viral infection. So we normally take vaccine before the infection, but if we have or if we acquired an infection, we must administer antiviral agents, suitable ones. Okay. Treatment of viral infection is difficult. Why? So in general, we are talking about the virus as a hard target. So هو الهدف الصعب للعلاج. If compared with the bacterial infections or the fungal infections, it is very hard to tackle or to kill the viral cell. Why? Because first of all, virus is a multiplying within the host cell. So it is protecting itself from the immune system because it or it enter inside the host cell. The second thing, we don't have so many biochemical mechanisms or pathways inside the virus in order to target. لأنه احنا حكينا في البكتيريا we have so many pathways. We have folate, we have DNA biosynthesis, we have so many metabolic pathways and enzymes, receptors that we can target in order to kill the bacterial cell. And the same story is for the fungi. But the virus, we don't have so many targets, so many enzymes or receptors. Because most of the pathways, the viral cell relies or depends on the host cell in order to grow and multiply. But we have some limited targets for the antiviral agents that we are going to talk about in the next lecture. This is all about our introduction for the antiviral agents for today, and thanks for listening. Assalamu alaikum.